Here we are, Annotated Digestive Physiology number 6. We're going to be looking at the pancreas in this video. So pancreas, remember, is retroperitoneal. That means it's behind the peritoneum. Head, body, and tail, the three parts of the pancreas. It's both an endocrine and an exocrine gland. Remember, by definition, endocrine secretions, which are hormones, travel by blood. Exocrine secretions don't. They're going to get dumped into the duodenum. So don't mix those up. In past semesters, I've had people get this mixed up. The pancreas does not secrete any hormones into the duodenum. It secretes hormones into the blood, and the two hormones it secretes are insulin and glucagon. Those go directly into the blood, whereas the exocrine secretions, the enzymes, are secreted into the duodenum. Keep those straight. Don't mix those up. Secretes insulin and glucagon into the blood. 1,500 mils of pancreatic juice, that's a quart and a half of Gatorade, roughly, into the duodenum every day. Water, enzymes, zymogens, sodium bicarb. Other pancreatic enzymes activated by exposure to bile and ions in the intestine. So, water, enzymes, a lot of them are in the form of zymogens. Now, you should pause the video. Why do you suppose the pancreas secretes most of its enzymes as zymogens, which are inactive form? Pause and come back when you think you know. Okay, what if they weren't zymogens? What if they were in active form? The pancreas would end up digesting itself, just the same way the stomach acid makes the stomach digest itself. And in the duodenum, remember, it's the chyme from the stomach, which is heavily acidic. And so in order to try to keep that stomach acid in the chyme from digesting the duodenum, the pancreas secretes a ton of bicarb to neutralize the acid. See, it all makes sense. This all fits together. If you know the basic molecules and stuff, which I've been emphasizing and stressing, then it's easy to see why the organs do what they do. Pancreatic duct runs the length of the pancreas, opens at the hepatopancreatic sphincter, and the accessory duct opens independently on the duodenum. So ducts of the gallbladder, liver, and pancreas. I've noticed I start most of my slides with either so or and. It's really boring, I'm sorry. Bile goes from the little lobules in the liver down into the right and left hepatic ducts. This is a great diagram. You should be able to follow this. You should know all this already. Those join together to make the common hepatic duct. Those then form up with the cystic duct to make the common bile duct. Okay? Major pancreatic duct and the common bile duct combine to form the hepatopancreatic ampulla, the wide spot where the two rivers come together, and then they squirt their secretions into the duodenum through the little nozzle, which is the major duodenal papilla. And again, the accessory pancreatic duct has a minor duodenal papilla. That's only enzymes. Major duodenal papilla is going to be both enzymes and bile. The sphincter is the muscle that keeps it closed most of the time. You don't want to be dumping that stuff in the duodenum just any old time. You only want to dump it in there when there's food. Gallbladder and bile, let's take a close look at those. It's a sac on the underside of the liver, 10 centimeters long. Wish we could be together in class because I love to show people 10 centimeters. Those in Bio Academy for Bio 201 should remember that. Half to a full liter of bile every day. That's a lot of bile. Gallbladder stores and concentrates bile. So not only does it store the bile, but it concentrates it. That's why when the gallbladder releases bile, it does a really good job of emulsifying those fats. If you have your gallbladder taken out, the liver still makes bile, but the liver then has to produce bile in real time. You don't have this big concentrated mass of it in the gallbladder, so you have to slow down when eating fatty foods if you have your gallbladder out. Bile backs up into the gallbladder from a filled bile duct. So, in other words, as the bile flows down the two hepatic ducts into the common hepatic duct, down into the common bile duct, because the sphincter is closed, it's going to start backing up, and it backs up into the gallbladder. backs up through the cystic duct into the gallbladder. Between meals, bile concentrated by a factor of 20, so it becomes much more concentrated in the gallbladder. Yellow-green fluid is what bile is, containing minerals, bile acids, cholesterol, bile pigments, and phospholipids. It's basically just a big thing of fats. There's kind of like a principle. If you want to break down fat, use a thinner fat. I used to work on cars a lot. You get your hands all greasy. Sometimes I would take like vegetable oil from the kitchen, and that would get all the grease from the car right off. Soap didn't work nearly as well as the vegetable oil. Bilirubin, the pigment from hemoglobin breakdown. Intestinal bacteria convert it to stercobilin. Bile acids emulsify fats and aid in their digestion. So remember, bile doesn't digest, it emulsifies. It breaks big chunks of fat down into smaller chunks, but now that they're in smaller chunks, then the lipases can actually go to work on them. There is what's called an enterohepatic circulation. Just look at that word. I keep emphasizing the breakdown of words. Look at that. It tells you exactly what it means. Entero means intestine, hepatic means liver. So what happens here is you've actually got blood then flowing from the intestines back to the liver through the hepatic portal vein. You actually recycle some of your bile. So you can reuse bile, all right? Now, in the pancreas, a couple kinds of cells. Acinar cells, they're called. These are the main cells that make the enzymes. Zymogens fall into several categories. So there are proteases. Proteases break down proteins. That includes trypsinogen, chymotrypsinogen, procarboxypeptidase. These are zymogens. That's so you don't digest your own pancreas. Trypsinogen will turn into trypsin. Chymotrypsinogen will turn into chymotrypsin. Procarboxypeptidase turns into carboxypeptidase. That's another way that zymogens are named, either with an O-gen at the end or with a pro at the beginning. So, look at pancreatic enzymes will break down proteins, okay? How many macromolecules are there in the human body? You should pause the video and try to remember for yourself. I'll wait. Okay, so we have proteins, lipids, carbs, and nucleic acids. Those are the 4 macromolecules. Let's see if the pancreas is going to be able to produce enzymes to take care of them. So far we're good on proteins. What comes next? Amylase. What does amylase break down? Starches. Remember, there was salivary amylase secreted in the mouth. Now this is pancreatic amylase. Make that chart, kids. So 2 kinds of amylase. Salivary and pancreatic. They're secreted in different places. They do their job in different places. They have the same function. Lipases digest fats. They break triglycerides down into glycerol and fatty acids. Ribonuclease and deoxyribonuclease, those break down nucleic acids. So remember, did we take care of all 4 macromolecules? We did. The pancreas makes enzymes that will break down any macromolecule that you can eat in your diet. And as I have emphasized before, the pancreas is what creates most of your digestive enzymes. Nice picture right here. Look at how things get activated. So, trypsinogen is converted to trypsin by enterokinase. That's an enteropeptidase. That's secreted by the small intestine. Here's an A's means it's an enzyme. It's an enzyme that trims the useless part of trypsinogen off, and then trypsin becomes activated. And then look what happens when trypsin becomes activated. I'm sorry, I went past that slide. Trypsin then will convert chymotrypsinogen into chymotrypsin, and it'll convert procarboxypeptidase into a carboxypeptidase. So notice again, as we saw before in the stomach, secretions affecting other secretions. Make that chart. Now, hormones. Secretin. Secretin is secreted by duodenal cells. It inhibits secretion of hydrochloric acid by the stomach. So, why do we do that? Well, homeostasis again. Homeostasis. If stuff is coming into the duodenum, it means the stomach has done its job. So let's tell the stomach to slow down. Stimulates the release of bicarb by the pancreas. And once again, bicarb is going to neutralize all that heavily acidic chyme that's coming in from the stomach. Also stimulates the production of bile by the liver. So look at the things secretin does, an incredibly important hormone for digestion. Cholecystokinin, CCK, released by duodenal cells, released in the small intestine again, just like secretin was. So remember on that chart, now we're seeing things that are secreted in the small intestine. Stimulates release of digestive enzymes by the pancreas. So in other words, how does the pancreas know when to release all those zymogens? Cholecystokinin tells it. Cholecystokinin, being a hormone, travels through the blood and tells the pancreas to start releasing enzymes. Also stimulates the gallbladder to release bile. Okay? Suppresses hunger in the brain. When we get to metabolism, we'll talk about this. You have various hormones that either make you hungry or suppress hunger. Cholecystokinin is released by the small intestine because, think about it, if you've got food in your small intestine, that means you're probably doing okay. So you want to suppress hunger so you don't overeat. Gastric inhibitory peptide, GIP. Its actual name was glucose-dependent insulinotropic peptide, but that was a mouthful. So they said, what the heck, let's make it simpler. And they found another way to do the acronym that made sense. Okay, is that cool or what? Hormone released again by the small intestine stimulates release of insulin. Why? Because if you're digesting food, you're going to create lots of new sugars. Well, that's going to raise your blood sugar. So what do you need to do? Release insulin. What does insulin do? Take sugar out of your blood and let cells absorb that sugar. Along with GLP-1, glucagon-like peptide number 1, constitute the incretins. So GIP and GLP-1 are the incretins. And look now, we've got secretins and incretins. Make that chart, boys and girls. You need to keep these separate. And they also inhibit gastric emptying and intestinal absorption. So again, what they're doing to inhibit gastric emptying, that means they're telling the stomach, slow down. We've got stuff down here in the small intestine. Take it easy for a little bit, okay? We're doing our job, all right? And so, one of my buddies, there is Heloderma suspectum, the Gila monster. I'm so sad. I was going to bring the Gila monster in for this class and let you guys all get to see it up close. So, why am I talking about Gila monsters? Oh, look, there's me giving a big, wet, sloppy one to the Gila monster. There's me doing one of my Gila monster demos, wearing a tie because it was a formal occasion. And there's me. Yeah, let's not talk about this. Used to give Gila monster weekly snake and Gila monster demonstrations at a park here in town. People used to take pictures and send them to me. I got like 850,000 of them. Okay, why Gila monsters? Type 2 diabetes? Because it relates to what we're talking to. This is so cool. Gila monsters may eat only 3 or 4 times a year. There might be a question or two on the exam about this because I think it's such a great story in science. Endocrinologist Dr. John Eng wondered how the hell can a Gila monster control its blood glucose? You have to eat a few times a day to control your glucose. How can a Gila monster eat only 3 or 4 times a year? Well, he discovered a compound called exendin-4 in Gila monsters. Isolated from the venom by Dr. Eng back in 1990, similar to glucagon-like peptide number 1, which we saw in the previous slide, okay? GLP remains active for only a few minutes. Exendin-4 remains active for many hours. So this was actually a medical breakthrough. By studying, here was somebody who's so clever, they started thinking about, how can Gila monsters do this? Then they went into the lab. They started looking at stuff, and by God, they found it. That's the way science is supposed to work. So what they did is they turned this into a drug called Enzymatic, sold as Byetta, for type 2 diabetics, all right? Used in conjunction with metformin, glucophage, helps control blood sugar in type 2 diabetics. A great success story involving my buddy, the Gila monster. And here's Kanab Point. This is my favorite place at the Grand Canyon. You can see for like 20 miles, both upstream and downstream. It's amazing. That's called the esplanade, all that orangish stuff. I think this is one of the most beautiful places on the planet. Okay, I'll see you again in Digestive PowerPoint Physiology number 7. Maybe. We'll see.